Okay, so we are talking about our macromolecules, right? We did carbs. All the way over here? Oh, never mind. Yeah, I guess I'm going to assume that we can see that on the screen. Carbs. Lipids. We did carbs and we did lipids, and what we're looking at is how we take monomers, small molecules, and we put them together to make big molecules. In just a second, we're going to talk about proteins. But and just a reminder, the monomers that we talked about for carbohydrates were the glucose and Glucose spells glucose, and the lipids, which were just sort of long carbon and hydrogen chains. Do they remember what these are called? The little things that go together to make lipids.
What's the first letter?
What?
What's the first letter? It's not a letter.
Good job.
No.
Fat.
Fruit roll-ups.
Fat is the first three letters of the actual word that this thing is. Yes.
Oh, are you asking us what lipids are?
I'm asking what the monomer is that goes together to make lipids. Did somebody just say?
Fatty acids.
Fatty acids. Thank you very much. That is correct. So you've got the simple sugar. Usually the example we use is glucose that are strung together to make various kinds of carbohydrates. You have the fatty acid. You have the fatty acid that goes to make lipids. And today we're going to talk about the monomer that makes up proteins. Does anybody from their reading remember what that monomer is that makes up proteins? That when we put many of them together, we get a protein. Oh, that's the other one. Actually, I appreciate that you remember that from the reading, but it ends with the same word.
Is it amino acid?
Amino acid was the word you were starting to say? Okay. All right. Good. I'm glad that was the... Anyway, all right. The amino acid. Yes. And I want you all to think back, and if you have to, turn back to the day when we started organic chemistry and we went through some of the functional groups, because amino acids, almost their whole chemical structure is right there in the title. There's a carbon in the center, and then there's, I need blue, hold on. Blue.
Oh, this is going to have to watch this.
So, amino acid is how this goes, and does anybody remember? So, and then there's the other. We'll fill in the rest of the carbon in a second, but the name really describes for us what goes into this. Does anybody remember what the amino group looks like? From
I wrote it down.
You wrote it down?
It's R.
Right. R meaning it could be attached to anything.
Yeah.
Yes.
R N H H.
NHH? NH2? Yes.
NH2.
N and two H's. Usually in the body, a third one is added on and it's called NH3+, but let's just do it like this because it's the amino group that we learned. And then on the other side, we've got an acid, and we learned what an acid is in organic chemistry.
What does the C mean?
The C is a central carbon.
Okay.
It's literally the very center of the, it's the chewy center. What about the acid? Does anybody remember what those look like?
Yes.
How about somebody else? You told us what the amino group is. All right, Cassie, what do you say?
It's R and then it's C,
Uh-huh.
double line O,
Double line. What does a double line
a
mean?
double bond,
Double bond.
and then on other different lines, OH.
OH. Yes. Okay. Yeah, that's an amino acid, except for two things. There's a... let's put it down here. Oh, yay, marker shirt. Look at that. I just drew right down my marker spot there. So sad. I'm a sad man. Okay. H, right? There's our little hydrogen there. And then we're going to fill in the rest. I need my purple marker. R. There we go. So R, everybody remembers what R is? What is R? When we see R in any chemical kind of thing, R means anything. R could be anything. I mean, not like a pony, but I want a pony. I want an Oompa Loompa and I want it now. Okay. Put a camera on me. This is what happens now. Anyway, so R can be anything, and this is the thing. If you line up all the amino acids in the world, and basically what happens is you connect a bunch of these to each other. By the way, you don't have to memorize this any more than when I drew out this full glucose molecule. You have to memorize like all those OHs and Hs and CH2OH and all that, or this whole thing. You don't have to. You'll have to recognize it. So let me put it over here. Protein C R H C O O H. That's the way you abbreviate this whole thing. When you just write it like, you can just write it like that, and you can write NH2. So yeah, you have to recognize that from that, like, but you don't have, I'm not going to be like, draw me an amino acid. That'll be something that you can do when you take, biochemistry in college or organic chemistry in college, which I recommend. But anyway, of course I would recommend that. What is the point? Okay, first of all, let's talk about a couple things having to do with proteins, a couple of general things. I call proteins globs with jobs. It's true.
Abbreviation or?
No, but because proteins actually do stuff. Proteins are the things in our bodies that move, that open and close, that actually do things. If you think about what all the other things do, what do lipids do? Well, they pretty much just sit there and they store energy for me. Carbohydrates, generally I use them for energy. They have other functions in cells, like to help differentiate one type of cell from another type of cell, packaging sort of stuff. But mostly they're also static, meaning they're not doing anything. Whereas proteins are the things that are doing things, like muscles. When you contract a muscle, that's a protein that's changing shape. Proteins do all the stuff that changes and moves, opens, closes, that kind of stuff. When proteins tend to be the things that, well, like a lot of our hormones are proteins, but not all of them. I shouldn't get too much into that. But like insulin is a protein. Insulin has a shape that's very specific, and the way they get these shapes is you string a whole bunch of these together, and what happens is that they take on different shapes because of this. With that said, I want to move over to here. Can you do me a favor? Can you make that shine on me yet again while I get this going?
Maybe. Possibly this one is different.
Oh, you don't have to take, you could just move, can you move the whole thing over? Yeah, that's fine.
Better.
I'll tell you what, I'm going to turn this on and we'll see if we can actually see what's on the board. I don't know if it'll be visible or not, but we'll give it a shot. I'm going to turn this off.
Every single time.
Like, why did you start doing that?
That poor bird.
It's a light.
Did you put it in the corner so that it couldn't see people moving?
No, it's not the speed to see people moving. It's
Are you going to
every
stuff
time
it
the light
in
goes
your-
off.
Can you do me a favor, Morgan, and just see if that actually shows up at all?
Mr. Arbach, are we writing?
No, you don't have to write all this down.
A little bit, just like really white.
Yeah, I'll tell you what, I'm just going to take it and I'm going to put it over here. Hello, I'm Mr. Auerbach. I will be your science teacher today.
Smash that part. Oh, that's some great filming right there.
That is what I am talking about. That is what I am talking about. Why won't you?
Let's not make the viewer motion selection.
The viewer is probably already somewhat sickened to begin with.
Wait, who's gonna look at this?
What's that?
Who's gonna look at this?
Who isn't? I've got at least a dozen hits on some of my videos.
Oh, there's more.
Anyway, okay, so all right, this horrifying mess of biochemistry is actually all of the amino acids. Let me shrink it so you can see them all. Whoa, there they all are. But let's make it larger so that you can actually see them. Okay.
Oh my god.
Okay, so what I want you all to see, just for a second, I'm not going to focus on this for very long. All of these are amino acids, and here is, it's like you took what I drew over there and turned it 90 degrees, because now here's your central carbon here, here's the amino group, here's the acid group. Notice that it's a little different. They put an extra hydrogen on there, which gives it a plus. They took away the hydrogen over here, which gives it a minus, but it's still, that's the COOH, that's the NH2, slightly modified. Here's the carbon with its extra hydrogen, and then this, in blue, is R. So what's in pink on this entire thing is always the same thing. Notice it's always, that's your amino acid backbone. Backbone's not the right word, but I can't think of the right word, so I'll just use that. Skeleton, fine. This is the R group. Notice how different the R groups could be. CH3, and then this has two CH3 groups on a central carbon, and then another carbon, and yet again. And then we get into the really weird ones. Phenylalanine has a CH2 with a benzene ring attached to it, a six-part carbon ring with 3 double bonds. Here's tryptophan. Tryptophan's the stuff that makes you fall asleep after your turkey dinner. Here's methionine. That has a sulfur in it. They're all very different. And what I want you to notice is that they all do different things based on their charge. So the last thing I want you to notice about these is this. This whole bunch that I just showed you are nonpolar, meaning that the R group, just CH3s, just like our fatty acids, it's all carbon and hydrogen, they're nonpolar. Over here, these are all polar. I want you to notice the presence of oxygen here, which is an electron-rich environment that's going to create a separation of charge. Sulfur will do the same thing. Sulfur and oxygen are in the same chemical group, and so they will act the same way. And then this OH, again, this nitrogen, also the nitrogen and the oxygen will cause a separation of charge. And so those are very polar. Amino acids that are polar and nonpolar are going to act a different way in the water environment that they exist in. The last thing I'll show you is that some of them are acids and some of them are bases. And no, you don't have to write these down. I think somebody asked me that. But notice how this is our R group. You already have your acid group over here, but this one has its very own acid group. There's the C, double bonded oxygen, and an O. And this is this can also be with the H on it like an acid. And then here's another one. And then these are bases. This one you sort of have to take on faith. That's a base, because we've talked about bases that have like an OH group on them, but NH3 plus is ammonia, and that's a base also, as we saw in our acid-base demonstration. So these NH3 pluses also act like bases. So you have amino acids that are polar, amino acids that are nonpolar, amino acids that are acids, and amino acids that are bases. And what does that mean? This is the last thing before we get up and do something.
Oh gosh. Why would you call an amino acid that is a base an acid?
That's a good question. Well, because the amino acid structure itself
Is an
has the amine group and the acid, the amino group and the acid, and then the R group happens to be a base. It's a good question. It's like, it doesn't neutralize itself or anything like that. It doesn't quite work like that. It's just sort of one of those things that you just kind of have to take it on faith that it is. It's like, okay. It's like being a Democrat in a state that votes Republican. It doesn't negate you as a Democrat. You're still there. You're still a Democrat in a state that votes Republican. Does that make any sense?
Yeah, sure.
Thank you for saying yes. Whoop, wrong one. Oh no. Oh, here it is. Okay. So the very last thing I want to show you is this is your average protein here. And notice this is why I mean by globs with jobs, because each of these spheres is a different amino acid. This protein has probably hundreds of amino acids. And the thing about them is, I don't want to listen to this person talk, so I'm just going to hit play so you see the next kind of picture here, because they basically say what I just said, except probably better. And so, no, we don't need next because all we need is this. What we're about to do, we're going to get up and we're going to go to the lab area and we're going to do an activity where we see how we go between this, which this is the polymer. You take amino acids, each of these little spheres is that thing I showed you over there. Tick, 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 tick. They're all in a line, but the thing is, they actually end up shaped like this. And that's because you take this and you put it into water
Oh, I get it.
and it's going to act, it's going to get all weird because of the way that the individual amino acids interact with water. And believe it or not, if you take, let's just assume that this was the line of amino acids that gave this, even though this is obviously 100 times shorter than it would have to be. But if you take this and straighten it out into a line, put it into water, more or less, it's going to make the same shape every time because of the stuff that's on it. And that's the cool thing about proteins, is that their shape is derived from the actual order of the and choice of amino acids that go on there. Out of the, there are 20 different amino acids, and out of those 20, you might have, you might have all of them represented, you might have half of them. There's no rule, because they all do different things, and they make different shapes depending on what they do, depending on, I'll edit this part out, depending on blah, 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 blah. Thank you very much. I am Mr. Auerbach. Okay, so we're going to do a thing now and go to learn about protein shapes.
Okay.